Welcome back. This is Muscle Physiology Part 2. If you haven't watched the previous video, Muscle Physiology Part 1, please go back and watch that first before coming back here to watch this one. So in the last video, we talked about how the signal to contract comes down the motor neuron, splits into a number of different terminal ends, and then a bunch of different things have to happen. A lot of molecules and ions have to move across the space, the synapse between the motor neuron and the muscle fiber. And all of the things that we talked about resulted in the release of calcium. So that increase in what we call intracellular calcium, the calcium that's inside the sarcoplasm, is going to do something to allow muscle contraction. And in this video, we're going to look more closely at exactly how that happens. Let's zoom in. Now we're getting very, very small. So before we can talk about exactly what calcium is going to do, we have to talk about what's inside these muscle fibers. There are long protein fibers. One of them is called actin, and one of them is called myosin. And myosin is the thick filament. And I'm going to just draw a little piece of myosin right here. And it forms a structure with actin that's almost like a little sandwich. And actin has pieces that are kind of coming in there, and you can see it's much thinner than our myosin, and it's sort of sandwiched in there. And this little section that we're drawing right here is called a sarcomere. And this is the section that actually gets smaller during a muscle contraction. And a sarcomere goes from one Z line, oops, that's not very straight, to another Z line on the other side, also not very straight. So these guys are called Z lines. And the myosin, an actin, is attached to the Z lines on either end. Now, normally, myosin has these little heads that stick out, almost like little hands, and it wants to bind to actin because it loves actin and it wants to grab onto actin and pull it. But it is prevented by a molecule called tropomyosin. So tropomyosin makes a little block here where it doesn't allow the myosin heads to bind to actin. So remember, myosin is the thick filament, actin is the thin filament, and the myosin heads are prevented from grabbing on and binding to actin by tropomyosin. And tropomyosin is associated with another molecule called troponin. Now, when calcium is low in the sarcoplasm, this is the setup. No contraction is happening because there's no calcium. But once calcium comes in, it's actually going to bind to, that molecule, troponin. Troponin is going to move out of the way because once it binds to calcium, calcium is a positively charged ion, it's going to change its shape. And when it moves out of the way, it's going to pull tropomyosin out of the way as well. So what's going to happen is when calcium is in the sarcoplasm, it gets pulled away from blocking these sites. It gets pulled sort of away from them. So this is tropomyosin and troponin when they're bound by calcium. Calcium causes them to move away, and now myosin can do what it wants to do, which is reach up and bind to actin. And each of these little heads is going to reach up and bind to actin. And when they do that, because the heads have energy in them, they're almost like a rubber band that's been pulled back. As soon as it can, that head is going to move forward and it's going to pull actin in this direction. And on this side, we're going to reach up, we're going to bind to actin, and we're going to pull actin in that direction. So in this manner, the sarcomere, which is from Z line to Z line, is going to shorten. That side will come this way, and this side will come that way because actin is being pulled that way, and it is attached to the Z line. So our sarcomere shortens whenever calcium is available. It pulls tropomyosin and troponin out of the way, and myosin can bind to actin. Now, that is called the power stroke. So these guys are all ready to go, and as soon as they can, they will go from this position back here and pull. Oops, that's the wrong color. And they will spring forward, pulling actin with them. So they kind of flap forward. It's almost like a reverse tug of war. So normally in tug of war, you would be pulling on either end of a rope and trying to pull the other team one way or the other. In this manner, each of these myosin heads are going the opposite direction. They are trying to pull the ends towards the middle. And by going hand over hand, they're going to shorten the sarcomere. Once calcium leaves, so when we get rid of calcium, let's erase our calcium from coming in, it's no longer bound to troponin and tropomyosin. And so troponin and tropomyosin will actually come back in here, and they will block those binding sites again. So instead of being out of the way and bound to calcium, when they're free, they will come back and they will prevent myosin from binding to actin again. So in this way, the presence or absence of calcium regulates whether or not muscle contraction will happen. And you have to realize that this is just one sarcomere that I'm showing you. In reality, there are thousands and thousands of sarcomeres all in a line, all attached to each other. And each one, these aren't exactly the same colors that I'm using, each one, as myosin binds to the actin and pulls it, is going to shorten. So this is the next sarcomere down I'm drawing. Those are my thick filaments, myosin. Thin filaments are actin. I always remember because actin sounds kind of like active. So anyway, so this is how this all works. And so really, all of this depends on the calcium concentration. When calcium is high, we get muscle contraction. When calcium is low, troponin and tropomyosin are blocking those myosin binding sites on actin, and muscle contraction can't happen. So if we zoom back out again, anything that interferes with calcium getting out of the SR or causes too much calcium to get out of the SR is going to cause problems in muscle contraction. And whenever we talk about problems with a process, we call it pathophysiology. So physiology, we already know, means how it happens, and patho means there's something wrong. So pathophysiology is there's something wrong with what's happening. And so there are a lot of different things that can disrupt this system. Let's look at some of them. So for example, let's say something happens here with the action potential. It cannot reach the terminal end of the neuron. So there's something wrong with the neuron. So sometimes the signal doesn't get sent. So for example, in Parkinson's disease, there's a problem with the neurotransmitter dopamine. It doesn't make enough dopamine, and so a lot of signals just don't get sent appropriately. Or in multiple sclerosis, the immune system actually attacks the myelin sheath that's around the neuron and causes a degradation of the signal. And so the signal never really gets to this terminal end. The signal never gets to the terminal end, we don't have an action potential to open these calcium gates, and we will not release the acetylcholine, and then the muscle cell will never even get the signal to contract. You could have problems with acetylcholine. Maybe you don't make enough acetylcholine. Or, for example, you have acetylcholine, but for some reason, these vesicles can't fuse with the membrane of the terminal end. And so acetylcholine never gets out. Again, that would result in paralysis. You would not be able to contract the muscle. You could have a problem with the muscle cell itself. So in muscular dystrophy, that's a genetic disease where there's a protein that helps to hold the muscle fiber together and keep it intact. And that protein is defective in people with this disease. And so maybe you can't maintain the gradient of sodium ions or calcium ions because the cell is damaged. So you might be wondering, why are we looking at pathophysiology? We are not going to be diagnosing patients. The reason we study pathophysiology is to help us better understand and assess our knowledge of normal physiology. For example, if I asked you what would happen if acetylcholine did not get released from the terminal end of the neuron, and you were able to tell me exactly what would happen and what that would mean for muscle contraction, that means you know pretty well what the normal physiology is supposed to be. So it's a way to really assess where you are and see if you understand normal physiology. The answer to that question would therefore have to be very specific. You couldn't tell me it just wouldn't contract because that's not specific enough. What I'm looking for is that ACH would not be able to diffuse across the synapse and therefore couldn't bind to the receptors. And so no signal would get from the neuron to the muscle fiber and contraction couldn't happen. That shows me that you understand what is normally supposed to happen and that it is not happening in this case. Okay, so we need to also talk about how to stop muscle contraction. In order for muscle contraction to stop, we have to stop the action potential coming down the motor neuron. Once that stops, we no longer release acetylcholine into the synapse. And once that stops, we no longer send a signal to the muscle fiber. The action potential then stops on the muscle fiber, and calcium is no longer released from the SR. And when intracellular calcium in the sarcoplasm falls, we know that actin and myosin can no longer contract and they start to slide apart. So all of these things have to happen to get rid of muscle contraction. Now remember, we move a lot of things around in muscle contraction. We're using a lot of facilitated diffusion where things are moving down their gradient and causing electrical signals. One of the reasons we use so much ATP in muscle contraction is because we constantly have to put them back. We have to transport or pump them back against the gradient using active transport. For example, when calcium enters the cell at the terminal ends, we use facilitated diffusion. And to get it back out again, we use active transport to move it against its gradient. Likewise, when sodium enters the muscle fiber, we use facilitated diffusion to get it in there. We will pump it back against its gradient by active transport to help maintain that gradient. But of course, that also uses ATP. And calcium leaves the sarcoplasmic reticulum by facilitated diffusion, and we have to always make sure we're putting it back into the sarcoplasmic reticulum by active transport. That's it for today. See you in class.